Okay, let's jump right in. Ever find yourself trying to get your head around something really complex, maybe something about a health issue for someone you know,
Mm-hmm.
and you hit the research papers, and suddenly it's just jargon everywhere. Feels overwhelming, right?
Yeah, absolutely. And that's kind of what we're trying to cut through here.
Exactly. Taking those complicated subjects and pulling out the key bits, the things that really matter, so
Yeah.
you get the picture without drowning in detail.
Making it click, essentially.
Precisely. And today, we're wading into, well, a really fascinating and vital area, Alzheimer's and the Parkinson's disease research.
And we're focusing specifically on the insights from Professor Chen, who is a major figure in this field based at Harvard Medical School.
Right. Our main source, our sort of stack for this deep dive, is her big plenary lecture at the ADPD conference.
Which was a pretty significant one, wasn't it? It marked the conference's 40th anniversary.
And her 30th year researching Alzheimer's. So a really key moment to capture her perspective.
Definitely a milestone moment. 30 years is a long time in research.
So our goal, our mission for you today, is to basically trace Professor Chen's path in this research.
Uh-huh.
Look at the big discoveries from her lab about the genetics behind Alzheimer's and Parkinson's, and, figure out what it all might mean for new treatments down the line.
And we're going to try and do that without making your head spin with technical terms, just the crucial, interesting stuff.
The real nuggets.
Exactly. It's interesting to start with her journey. She mentioned her Alzheimer's work kicked off 30 years back at MIT.
She was drawn to the genetics of it all, right?
Yeah, apparently she was in a lab studying learning and memory initially, so the link to dementia, to that cognitive decline, became sort of a central thread for her.
Makes sense. And she really honed in on those causative genes in familial Alzheimer's PS1 and PS2.
And it's important, like you said, to distinguish those from risk factors like APOE.
Because these causative ones, if you have the mutation, your chance of getting Alzheimer's is extremely high. Highly penetrant, as she put it.
Right. And this ties into the whole history, the amyloid hypothesis that's dominated for so long.
The idea that amyloid protein buildup is the main problem.
Yeah, finding the APP gene connection in 91 really fueled that. And then finding that presenilin mutations changed the ratio of amyloid types.
A42 to A40.
Exactly. That seemed to cement the amyloid idea and led to, well, decades of trying to target amyloid.
But Professor Chen's lab took a slightly different angle, didn't they? Really asking, what does presenilin itself actually do?
And that's what led them, back in 2006, to formulate the presenilin hypothesis.
Okay, so what's the core idea there?
In simple terms, it's that losing the normal function of presenilin, just the protein not doing its usual job properly, actually contributes directly to the nerve cell damage and the dementia.
Not just its role in making amyloid.
Precisely. And crucially, their hypothesis predicted something important.
What was that?
They predicted that drugs simply blocking gamma-secretase, the enzyme complex presenilin is part of, would probably make things worse for patients.
Which, unfortunately, turned out to be true in some clinical trials later on.
Sadly, yes. So, to test their own hypothesis, they did a ton of experiments.
Using cell cultures, biochemical systems.
And importantly, these sophisticated mouse models. And the evidence started piling up.
Showing what, exactly?
That when presenilin function is knocked down by these Alzheimer's mutations, these loss-of-function mutations, it messes up gamma-secretase activity, yes, but also learning, memory, and crucially, neuronal survival. The nerve cells themselves struggle.
Wow. So it's hitting multiple systems.
It seems so. And they even had some early preclinical proof-of-concept work suggesting gene therapy might help.
In mice carrying those presenilin mutations.
Yeah, introducing a healthy copy of the gene. It was an early hint that directly boosting presenilin function could be a therapeutic avenue for those specific genetic cases.
Okay, so that's the hypothesis.
Yeah.
But then they also dug into what presenilin normally does, right? Beyond just chopping up APP.
Absolutely. And that work was really revealing.
What did they find?
Using both mice and, interestingly, fruit flies, they showed presenilin is vital for really basic fundamental stuff like synaptic function, how neurons talk to each other, also learning and memory, and just basic neuron survival. It has a much bigger day job than just processing amyloid.
And didn't they find something quite unexpected when they specifically switched off presenilin just in certain brain cells in mice?
Yes. That was a key experiment. They inactivated presenilin only in the excitatory neurons of the mouse cortex, the outer brain layer.
And unlike just having too much amyloid, which doesn't necessarily kill those neurons in mice.
Right. In this case, losing presenilin function led to progressive neurodegeneration. those cortical neurons started dying off.
That really points to presenilin itself being critical for keeping those neurons healthy, doesn't it?
It strongly suggests it, so then they asked, okay, how? What's the mechanism?
And they went back to the fruit flies for that.
They did. Genetic screens in Drosophila are powerful for this. They found genes involved in lipid metabolism, how cells handle fats that interact with presenilin.
Lipids. How does that connect?
Well, when they reduced presenilin function in the flies, messing with these lipid genes made the neurodegeneration worse.
Ah, okay. So there's a link between presenilin and how brain cells manage fats.
It seems so. The idea is that when presenilin is faulty due to these mutations, it's not just messing up APP processing, it's also disrupting these crucial lipid pathways inside the neuron.
Contributing further to the cell's problems and eventual death.
Exactly. It reinforces that loss of function isn't just about one pathway. It has broader consequences within the cell.
Okay, so we understand presenilin's normal jobs are important. The next step is figuring out how these tiny genetic changes, the Alzheimer's mutations, actually break it.
Right, because often these are just single amino acid changes. Very subtle.
So what did their analysis show? Are most mutations gain of function or loss of function?
The overwhelming majority they tested were loss of function. They either reduced or completely wiped out gamma-secretase activity.
And they used those knock-in mice again to study specific mutations.
Yes, models with mutations like L435F and C410Y, which are known to cause aggressive early-onset Alzheimer's in humans.
And what did those mice look like?
Quite striking, actually. They showed features very similar to mice completely lacking the presenilin gene.
Even though the mutant protein was still being made.
Yeah, it seemed like the mutant protein was there, but it wasn't getting processed correctly or wasn't assembling properly into the gamma-secretase complex. Its functional output was severely reduced.
And did the severity in the mice match the human disease?
Generally, yes. The mutations causing more severe deficits in the mice tended to be associated with more aggressive disease in humans.
And then the structural biology work came in later.
Exactly. Studying the 3D structure of gamma-secretase actually showed why some mutations are so bad.
Like the L435F one.
They found that mutation sits right in or extends into the enzyme's active site, the critical working part.
So a structural reason for the loss of function.
Precisely. It provided really strong validation at the molecular level.
Okay, but here's perhaps the most critical finding, the one that really challenges the amyloid-only view. Tell us about the experiments crossing these mice.
Right, this was crucial. They took their presenilin knock-in mice, the ones with the loss-of-function mutation, and bred them with mice that were engineered to completely lack the APP gene. No APP means absolutely no amyloid beta can be produced, zero.
So the question is, if there's no amyloid, does the presenilin mutation still cause problems?
And the answer was a clear yes. Even without any the mice with the presenilin mutations still developed neurodegeneration.
The brain cells still died.
Yes. The loss of brain tissue, the neuronal death, it still happened. Removing amyloid didn't prevent it.
Wow. And what about learning and memory?
Still severely impaired in the presenilin mutant mice, even with no APP or AP present.
So presenilin's role in cognition is also, at least partly, independent of amyloid.
That's what the evidence strongly suggests. It indicates that presenilin is doing something fundamental for neuron survival and function that goes beyond its role in AP production.
This is a really major point for you, the listener, to grasp. While amyloid plaques are a hallmark of Alzheimer's, this work shows that problems with presenilin itself can drive the disease process, even without the amyloid.
It fundamentally broadens the picture. Presenilin isn't just a supporting actor in the amyloid story. It seems to have its own leading role in neuronal health.
Okay, so how does this compare to mutations in the other major familial AD gene, APP?
That comparison is also quite telling. When they knocked out the entire APP gene family in mice, so not just APP, but its relatives too.
Did that cause neurodegeneration like the presenilin loss did?
No, at least not the same kind of cortical neuronal death. Those APP family knockout mice had issues with learning, memory, and synaptic function, yes, but not the widespread cell loss seen with presenilin inactivation.
Interesting. So losing APP function seems less detrimental to basic neuron survival than losing presenilin function, at least in these models.
It appears that way, and Professor Chen also highlighted a difference in where the mutations occur.
You mentioned this earlier, APP mutations cluster.
Right. They tend to clump around the site where atin is cleaved off the main protein. But presenilin mutations are scattered all over the gene.
Does that potentially explain the different outcomes?
It might. The widespread nature of PSNI mutations could suggest they disrupt multiple aspects of presenilin's function or structure, whereas APP mutations might more specifically affect ANF production. It's a plausible explanation for the phenotypic differences.
And what about the human data? Does it align?
It does seem to. Large studies show that people carrying mutations in PS1, which is the most common familial AD gene, generally get the disease earlier than people with APP mutations.
Suggesting that PS1 mutations might be initiating a more severe or rapid disease process.
Potentially, yes. It adds weight to the idea that presenilin function is incredibly critical, and losing it has profound consequences, perhaps more direct or immediate than altering ATMR production alone in some cases.
Okay, so we've built this case for presenilin's crucial role and how losing its function drives disease. Let's pivot to the potential upside. The therapy. What about that gene therapy idea?
Right, the preclinical work they did, it's based on this loss of function concept. If the problem is not enough functional presenilin, maybe adding some back could help.
So they tested this in the mice with that severe L435F mutation.
Exactly. They used a gene therapy vector, basically a way to deliver genetic material to introduce the normal, healthy human PS1 gene into the brains of these mice.
And did it work? Did the healthy gene take hold?
It seemed to. They saw evidence that the introduced human presenilin could actually incorporate into the gamma-secretase complex and even displace some of the faulty mouse protein.
Okay. And did that restore the enzyme's function?
Yes. One key sign was a reduction in the buildup of a fragment called APP-CTF.
Remind us what that is again?
It's a piece of the ATP protein that gamma-secretase normally cuts. If gamma-secretase isn't working well because of presenilin mutation, this CTF fragment accumulates.
So less CTF means the enzyme is working better.
Precisely. The gene therapy was helping to clear that backlog.
But here's the really striking part. The amount they needed.
This was the kicker, yeah. They found they didn't need to restore presenilin levels back to 100% of normal.
How much was enough?
Expressing the healthy human PS1 at only about 20% of the levels seen in normal wild-type mice was sufficient.
Just 20%. And that was enough to make a difference.
Apparently so. That modest level restored gamma-secretase activity significantly. It improved the mice's performance in learning and memory tests, and critically, it reduced the amount of neurodegeneration observed in the brains.
That's actually incredible. Just a relatively small boost in function had major benefits.
It really is. It suggests there might be a therapeutic window where you don't need full restoration. Just getting function back above a certain critical threshold could be highly beneficial.
So this offers a potential pathway for treating people with these specific presenilin mutations.
It certainly provides a strong preclinical rationale. It suggests that gene therapy aimed at providing even a modest amount of functional presenilin could be a viable strategy for this subset of Alzheimer's patients.
Obviously, translating that from mice to humans is a huge leap, but the principle is powerful.
Absolutely. Many hurdles remain, but it's a very promising direction based on understanding the fundamental biology of presenilin loss of function.
And are they continuing to test this hypothesis? They may be looking at APP mutations in a similar way?
Yes, Professor Chen indicated they are continuing to test the boundaries of the hypothesis, including using similar unbiased genetic approaches to dissect the effects of APP mutations as well. The story is still unfolding.
Okay, so let's try and wrap this up. The big takeaway from Professor Chen's work seems to be this compelling case that presenilin not working properly, this loss of function, is a really key problem in familial Alzheimer's.
And critically, that this problem can drive neurodegeneration and cognitive issues even independently of amyloid beta buildup.
Which definitely shifts the focus, or at least broadens it, from just targeting amyloid.
It adds a major piece to the puzzle for sure. And that aha moment, I think, is the gene therapy result.
Right. The fact that introducing just a little bit of healthy presenilin could rescue function and reduce degeneration in those mice.
That's pretty remarkable and offers real hope for a potential treatment approach for those specific genetic forms of AD.
Absolutely. So for you listening, we hope this journey through Professor Chen's research gives you a much deeper appreciation for the intricate genetics of Alzheimer's.
And a window into how this kind of fundamental research, really digging into what these proteins do, can point towards completely new ways to think about therapies.
It really underscores the complexity, doesn't it?
Yeah.
And maybe leaves us with a final thought to chew on.
What's that?
Well, if presenilin, often seen mainly through the lens of amyloid, turns out to have these critical independent roles, what other molecules in other complex diseases might we be underestimating?
That's a great point. What other secondary players might actually hold central keys if we look beyond the dominant hypotheses?
What assumptions might need a second look in other areas of research? Liking to think about.